Hi, welcome to Botany 101. I'm Angela Nishimoto, okay? And today we're going to do 3 different things a little bit, okay? First of all, we're going to go over your first assignment, all right, which is a writing assignment, all right? Second, we're going to do a little mini review of some of the features of the cell that we talked about last time, just a couple of them. And then thirdly, we're going to continue with our look at the plant cell, okay? Adventures. So your first assignment is called Learning Log 1, and it does have to do with a cell, which we've been learning about last time, okay, and we're going to continue this time, okay. For one thing, it's going to be a paper, a short paper, but it is a formal paper, okay. Short paper, 1 to 1.5 pages long, all right. So what we're doing is you are going to be taking 5 parts of the plant cell. Okay, you're going to be explaining what these 5 parts do, all right, and you're going to construct an analogy. You're going to actually explain, okay, how the 5 parts of the plant cell work to make the cell, the plant cell function as a whole, similar to the parts of one other system or the components of one other system. For example, okay, you could talk about a restaurant. Say you know how restaurants work. Yes, so say you actually work at a restaurant. You're thinking, this is something that you think you could do. For one thing, you could say the outermost part of the plant cell, the cell wall. You might be able to liken it to the infrastructure of the building, like the walls of the building that surrounds the restaurant. Okay. Secondly, another thing we talked about was the plasma membrane or the cell membrane. You can mention that maybe a restaurant has a hostess or a host or maitre d', and what that person does is they actually allow people in or allow people out. They kind of govern and kind of make sure who goes in, who goes out, if you're okay, and that everything is right. So they're sort of like a sentry in a way. Okay, so in this way, you're going to compare 5 different parts of the plant cell to 5 parts of some other structure, okay, or some other system. And a restaurant analogy might work. Another thing might be, comparing the plant cell parts to the parts of a computer. So one of the things they say is that if you have to write something, write about what you know. Okay, so that's always the best thing to do. If you're an expert at something, you might try to make an analogy between that part, let's say an automobile, or say if you really know about the components and the structure of a house, okay? So those are some different kinds of subjects you might try to make an analogy with the plant cell. So today we're going to do a little bit review, and then we're going to continue and proceed with the rest of the plant cell, okay? Now the thing about this assignment also is that it's due 1 week from today, okay, from the date of the airing of this. Another thing that you should know about this assignment is you may turn in a hard copy if you want to, but I'm completely fine, with email submissions. Okay, what you might want to do is word process it in a word processing program, and then what you do is that you will actually send it as an attachment to me, and on my email that's on the syllabus, okay, that you should know. Okay, so you should have a copy of the syllabus by now. So you can send it to me via email as an attachment. If I have any problems opening it up, okay, I will email you back and see if we can kind of rectify that. All right. So hopefully you'll pay attention and you'll learn about the plant cell, and it will help you, okay, in terms of your paper. So this is a formal paper. You have to use good grammar, okay, proper sentence structure, okay, no colloquialisms that will be, sort of like inappropriate for a college paper, okay? So looking forward to receiving them from you. It's always interesting to see what people come up with. People are very creative. Students, are very creative, okay? Okay, so today we're going to go over a little bit about the objectives first before we launch into today's lecture, all right? So for one thing, okay, the first objective, we're going to try to answer and continue answering what the components of the plant cell are. What are they? Yes, you remember we talked a little bit before about the cell wall, the cell membrane, a little bit about the cytosol, not that much, but also about a couple of organelles, yes? So what are the other components of the plant cell? What are the functions of these parts? Because, of course, these parts of the plant cell do things. Okay, they allow for division of labor, a division of function within the unit known as a plant cell. Recall too that a plant cell is very, very small. It's only 10 to 100 micrometers, right? 10 to 100 millionths of a micrometer. Okay, very, very tiny, but still very complex. Okay. Furthermore, we're going to progress into something called the cell cycle. All right, and the cell cycle. Okay, what we're going to find out about it. What does it do? What happens during parts of the cell cycle? Then we're going to get into mitosis. Okay, what is mitosis? And then what are the phases of mitosis? Right? Furthermore, we're going to go into what actually occurs. What are the main events during the phases of mitosis? All right, what actually happens during the phases of mitosis? Then we're going to talk about what are the functions of mitosis for the plant. What does it do? You know, what does it allow for the plant to do during the lifetime of the plant? Okay? Okay, recall from the last lecture that the protoplast is a living part of the cell, and the protoplast is enclosed by a non-living part of the cell known as the cell wall. Recall? And recall too that the protoplast contains cytosol, okay, or fluid inside of the cell, so an aqueous fluid, water-based, but the protoplast also contains organelles, okay? And recall before that we talked about chloroplasts. Chloroplasts are the sites of photosynthesis, all right? So here I have a leaf, a single frond, a single leaf of mylia-scented fern, also known as Loae, yes? And if you look at it, of course, the leaf is green, yes? So chloroplasts are the little organelles that are inside of the cells of this leaf, again, in some plants in the stem as well, and the chloroplasts are the little organelles that do photosynthesis. Within the chloroplasts, you have the grana and the stroma, which we'll go into. Anyway, so here we have a green leaf, very green, does photosynthesis. So whenever you see green in a plant, you know the organ has something to do with making sugar from sunlight energy, the conversion of sunlight or radiant energy into chemical energy, all right? Now, if you look at the opposite side, this is a little bit off the topic, but I wanted to give you a little preview. On the other side, okay, of this fern frond, you can see that you have these little tiny kind of, well, kind of small, brownish or orangish dots on them. Now, these are called sori. Recall when we looked at a fern, the first session that we had, you saw the fiddlehead. Remember the fiddlehead, that little curled up, unfurled frond? And when it opens up, and as the leaf matures, what you'll have is that you'll have these sori. The sori, S-O-R-I, have to do with sexual reproduction in ferns. Okay, because you remember, ferns are spore-bearing plants. They reproduce sexually through spores, whereas angiosperms and gymnosperms reproduce via seeds. Okay? All right. So recall that the chloroplasts are where photosynthesis happens. Okay, and the chloroplasts are green also, and they're green because they have the pigment in them, right? So recall too, the chloroplasts are surrounded by a double membrane. All right, two membranes that surround the insides of the chloroplast. Okay, and recall too, chlorophyll is a pigment, okay, within the leaf. So we have the two different kinds of chlorophylls in terrestrial or land plants, chlorophyll A and chlorophyll B. We also have other kind of pigments known as carotenes or carotenoids. Okay, carotenes can come in orange, they can be red, okay, brownish in color, and they allow the plant to harvest light, other parts of the light spectrum, okay, not just what the green can harvest. And we also have xanthophylls, which are oftentimes yellowish or brownish pigments. Okay, recall too that the chloroplasts are comprised of the grana. Remember, the grana are stacks of thylakoids. Thylakoids are membranous, okay, components of the grana. All right. And then we have the stroma. The grana is a place where the light reactions happen, and the stroma is where the Calvin cycle happens. So photosynthesis happens in two parts. So we humans divide it into two parts. We call it the light reactions, which happen in the grana, and we also have the Calvin cycle that happens in the stroma of the plant. Okay? All right. Then here we have an image of the chloroplast. You can see here the double membrane surrounding the insides of the chloroplast, okay, the outer membrane and the inner membrane. Here we have the thylakoid, okay, a single thylakoid, okay, and these thylakoids are arranged in stacks known as the grana, okay, G-R-A-N-A. Yeah. So a single grana is called a single one of the grana is called a granum, granum. So here you have a stack of thylakoid membranes. Then you have sort of like the jelly-like matrix, or the jelly-like part inside of the chloroplast, interior to the membranes, and this is called the stroma. Okay. Okay. So remember that the grana is where the light reactions happens, and the stroma is where the Calvin cycle happens. All right. Recall too that you have other kind of plastids. So chloroplasts are organelles that are under the heading of plastids. We also have leucoplasts, such as this great, photograph, great image of a potato cell. Okay, this is a cell of a potato, the tuber. The tuber is actually an underground horizontal stem, okay, full of starch. Because remember that plants will store their excess food in the form of starch for the most part. Okay, so here we have a starch grain in a leucoplast. Okay, so leucoplast, very well illustrated here, and we call it chromoplast. So we know chloro refers to a yellow-green color, leuco refers to white or a whitish color, and chromo just means color. Okay, or colored. So a chromoplast is a plastid that generally has red organelles in there called chromoplasts, or they can have yellow, or they can have orange organelles called chromoplasts. Okay. Now another organelle that we looked at was the mitochondrion. Okay, plural mitochondria. This is where cellular respiration happens. Okay, or most of cellular respiration happens. So the mitochondrion, okay, has a double membrane also surrounding it. Okay, which might give clues, just like in the chloroplast, to its origin. All right. Now, the double membrane that surrounds the mitochondrion includes the inner membrane that has infoldings called cristae, all right? And it is on the inner membrane that you have the third part of cellular respiration happening, and that is called the electron transport chain, okay? The first part of cellular respiration is called glycolysis. That happens in the cytoplasm of the cell. The second part of cellular respiration happens in the matrix of the mitochondrion. Okay, we'll take a look at that in just a second. Okay, so here it is. Okay, first part of cellular respiration happens outside of the mitochondrion. That's called glycolysis. All right. The second part of cellular respiration happens in here in the inner mitochondrial matrix, in the inner part of the mitochondrion. And you can see here, you can see an outer membrane, and here you have the inner membrane with its infoldings, and it is on or within the inner membrane that you have the electron transport systems located. The electron transport systems are responsible for the production of most of the packets of energy, which is called ATP, okay? Adenosine triphosphate. We'll get into that a little bit later, so don't worry about this right now. But within the membrane, you have enzymes called ATP synthases, and these actually will produce ATP. Okay, we'll go into this in more detail, you can be sure, later on. Okay, let's see. Okay, now we're going to continue with other organelles. For one thing, we have an organelle which is known as the vacuole. Okay, you can think of the vacuole. I guess when you think of vacuole, you can think of vacuum. But of course, it is because when people first discovered the vacuole, they thought there wasn't really anything much in there. Okay, the central vacuole can be the largest structure in a plant cell, and this is a structure that is present only in plant cells. I guess maybe also in algae, not sure. But they're present in plant cells, but not in animal cells. Animal cells do not have a central vacuole. And within the central vacuole, you have liquid, and that's called cell sap, okay? And also, the central vacuole, okay, inside of the plant is surrounded by a single membrane, and that's called a tonoplast. Okay, you know that we scientists, we have a lot of different terminologies for all these different things, and you folks do have to learn this, okay? So we have the tonoplast, and then within the vacuole, you'll have different kinds of substances such as salts, all right, sugars, all in solution in that cell sap solution, proteins in there. You'll also have crystals, okay, as well as pigments. So, you know, one of the plants that we talked about a little bit before, which is a very important plant, okay, this of course is called a little piece of a plant known as kalo, also known as taro. This one here is very, very dark, okay, in terms of the leaf, yeah? And, you know, if you kind of look at it, taro is very important locally, very important plant culturally to the Hawaiian people, okay, very much. The Hawaiian people consider that the kalo plant is actually a relative. It is related to them. They believe that the first humans actually sprang, okay, from you know, when you get into the stories of the people, yeah. So just remember that the taro, very important, very interesting plant. You know, very much grown very well here in Hawaii. And if you look at this, look at it, you know, when you're looking at the anatomy of it, if you look at the back part of the leaf, because this is part of the leaf, this is also part of the leaf, okay? This part is called the blade, and this is the part of the leaf that does most of the photosynthesis, and this part here is called the petiole, okay, or the petiole. And this is the leaf stem. So one of the ways you can identify a kalo leaf or a taro leaf is that the petiole joins the blade sort of interior to the margin or the edges of the leaf, okay? Remember that the kalo plant is very important culturally to Hawaiian people. You know, it is esteemed above all other plants, all right? Very important to know that, okay? Since we're here in Hawaii, all right? Okie doke, let's see. So taro. So I showed you that because the taro itself, okay, has these crystals in it that are called calcium oxalate. So if you can actually eat all parts of the taro, you can eat the corm, of course, which is what people used to make poi with, yeah, using the poi pounder. Another thing is that people can eat the lua leaf, yeah, the leaf of the kalo. Generally, any parts of the taro that you want to eat, you should cook it for a very long time. You want to make it very, very cooked, because inside of the vacuole of this taro leaf, you might be thinking, what does this have to do with the vacuole? Well, it's because the kalo leaf has a vacuole that has these crystals within them, and these crystals are known as calcium oxalate. And if you haven't cooked kalo leaf or kalo corm long enough, these crystals will actually embed themselves in your mucous membranes. That happened to me. I went to a supermarket once, you know, very good, and they had some taro leaves there for sale. I thought, yes, you know, I should cook this up, and it's really good, very nutritious, has a lot of iron and other, you know, minerals and vitamins, very healthy for people, okay? So I did, I took it home, I cooked it, you know, for maybe about 15 minutes. I thought, oh, it looks done, it looks like spinach, so it's ready to eat. So I ate it, you know, and it was delicious, of course, and then a few minutes later, I started to feel all this tingling in my mouth and in my, you know, my lip and in my throat. It was actually because I hadn't cooked it long enough. I hadn't cooked the leaves long enough. So you do have to cook it for quite a long time. What happens then is that these calcium oxalate crystals will break down, and then they'll be okay to eat. This, what do they call it? Itchy throat, I think it gives you. Another plant, which is called Dieffenbachia, it's in the same family, the Araceae. This Dieffenbachia plant, okay, it's also known as dumb cane. The word dumb originally means that you couldn't speak, so they think that if they know that if you eat the leaves or parts of this plant, the same thing can happen. Your mucous membranes can swell up, and you couldn't talk, you know, when you ate that. So that's actually not so good, right? Of course, but they even say that your throat can swell up to the fact that you can't even breathe. You know, that's kind of consternating, of course. Very, very. You have to be wary. Yeah. Okay. Now another kind of inclusion that you can sometimes find inside of a vacuole of a plant are called anthocyanins. Anthocyanins, antho just refers to flower, yeah, and cyanin, of course, is a kind of pigment. Cyan is sort of like a bluish color, yeah, so the anthocyanins usually are purplish, or they're pinkish, or they're bluish, and they're oftentimes responsible for pigments, okay, in some parts of the plant. So here we have a very, very nice image, and you can see here, this is from onion, yeah, probably, it is red onion, you know, the one that you can buy at the supermarket with that nice kind of purplish, reddish, burgundy colored skin on it. So these anthocyanin pigments actually make it, you know, that color. So this is a photomicrograph under a compound microscope of onion cells, of the red onion. And here you can see the cell wall very nicely here. And it's just that the vacuole of some cells take up a lot of space. So this is why I guess the whole thing looks pinkish in color. Okay, so we'll be looking at cells of red onion in lab. Okay, and we'll actually be doing a little, sort of like a little mini experiment in terms of osmosis, which we're going to get into later on. Okay? All right, now further, so we just talked about the vacuole, yes, and some of its inclusions, including salts and etc. Now we're going to talk about something known as the ER, also known as the endoplasmic reticulum. Generally in class or in the exams, I usually just call it the ER, okay? But the ER, okay, can be sort of segmented or divided into what they call rough ER, okay? They call it rough ER because the surface of this membranous organelle, okay, remember we've talked about so far, we talked about the chloroplast, okay, we talked about the mitochondrion, we also talked about the vacuole, and all those three organelles are surrounded by membranes, yes, the chloroplast and the mitochondrion surrounded by a double membrane, and recall too that the vacuole is surrounded by a membrane known as a tonoplast, okay, so those are organelles that are surrounded by membrane. Right? Now the ER is a membranous organelle. That means the organelle itself is made up of membranes. All right? And the rough ER has ribosomes studding the surface of it. That's why they're rough, because the surface of the ER is bumpy or rough with ribosomes. And what do ribosomes do? Okay? Ribosomes produce proteins. They're actually a place where proteins are synthesized or assembled inside of the cell. Okay, so the rough ER with the ribosomes has this function of producing proteins. And proteins, if we recall from the lecture on chemistry, very important, okay, because they provide structure. They also provide transport, you know, allowing substances to go across membranes. They also function in some hormones, okay, are also proteins, as well as enzymes. You can't do any chemical reactions without enzymes. So enzymes are of paramount importance. So recall too that proteins are very large and very complex molecules, okay, with up to 4 levels of structure. Remember too that their subunits are called amino acids, okay? So this is why we need to have nitrogen. Okay, because recall the amino acid structure, you have a central carbon, you have a carboxyl group, you have an amino group, you have a hydrogen, and then you have an R group, yes? So remember, you have to remember these things because you will probably be tested on them, okay? Now we also have smooth ER as well. Smooth ER doesn't have ribosomes, and they call it smooth ER because they're not all bumpy like the rough ER is, okay? So these usually produce lipids. They also produce other substances, but one of their main products are lipids, okay? Another membranous organelle is called the Golgi apparatus. Sometimes people call them dictyosomes, okay? And they're named after a scientist named Golgi, okay? And so what the Golgi apparatus does, it will actually take substances produced by the ER, whether the rough ER or the smooth ER, and they'll either, what they'll do is they'll actually finish them, modifying them, packaging them, and then packaging them within a vesicle, and then maybe allowing them to be exported out of the cell. Because sometimes one cell can make a product, and then what it does, it is actually translocated or moved to another part of the plant. Just like in our bodies, you know, with our hormones and etc. Plants have hormones too. Hormones are substances that are made in one part, transported to another part of the organism, and then it has an effect in the other part of the organism, okay? So hormones, very important in plants as well as in animals. And of course proteins too. Enzymes are sometimes made by the cell, move from one place where it's made to another part of the plant. So it actually is transported or moved, okay, from one place to another, okay, in the plant, okay? Now here we'll have an image of the membranous organelles as well as some other parts of the cell, okay? So here we have, okay, we're going to get into this a little bit. This is called the nucleus of the cell, okay? And then what you'll see here is that these membranes here, all right, are studded with ribosomes. You see these little, depicted as these little light-colored dots here? So that's supposed to be the ribosomes. Ribosomes, remember, are places where proteins are assembled, and ribosomes can either be floating freely in the cytosol or the fluid inside of the cell, or they can be studying the surface of the ER, the rough ER, all right? Now here, this is smooth ER, okay? Smooth ER is a place where lipids as well as other substances are produced, okay? Now one of the reasons why, sometimes people build up a drug resistance, such as in antibiotics and such, is because your liver, okay, your liver will make more and more smooth ER, because what's happening is that your liver is one of your main organs. It helps to detoxify your body. So what happens is that when you're taking something like even antibiotics or other things, other sorts of compounds that are chemical, what happens is that your liver will produce more and more ER. So what that does, or smooth ER, that produces substances that help break down these toxins in your body. Okay? So even antibiotics, even though they help you get over an infection, they will actually, your body perceives it as a toxin. So sometimes if you take antibiotics, if you have a tendency to get, lung infections, like not pneumonia only, but also others, that, bronchitis, or you have throat infections and such, that sinus infections that you need to take antibiotics for. Oftentimes your liver will make more smooth ER, so it can help to break down these toxins. And then what can eventually happen is that your resistance, or your the level of antibiotics or other kind of medications will actually get higher and higher. You need to take more and more to be able to get the same effect from it. Okay, so one of the things that are current now in medical practice nowadays is a lot of physicians and MDs, osteopaths, they're not really prescribing as much antibiotics as they used to before. Because for one thing, toxicity, and for another thing, the bacteria that you're trying to fight off will actually sometimes have resistance to these antibiotics. Okay? All right, so here we have the rough ER, here we have the smooth ER, and then here we have the Golgi apparatus. So what happens is substances made in the ER, whether the rough ER or the smooth ER, are enclosed within vesicles. Okay, this is like a little tiny little membrane, small little organelle with a membrane surrounding it, and then it actually will move through the cell and then attach to one side, okay, of the Golgi apparatus. So you can think of the Golgi apparatus as being almost like a stack of pancakes. Yeah, all these membranes, they're like pancakes, and they're kind of being produced or put together from one side and then consumed or eaten up on the other side. What happens is that the substance actually moves through the stacks, goes from one into the next and next layer, and then what happens is that it's pinched off, part of the vesicle, the membrane, makes another vesicle, and then they're exported out of the cell if that's what the substance needs to do. Okay, so here we have the nucleus, here we have the rough ER, here we have the smooth ER, and here you have the Golgi body, okay, or the Golgi apparatus, or the dictyosome. I know it's kind of confusing. You have all these different words, for the same thing. All right? Now another component of the cell, which we're not going to get into that much here, is called the cytoskeleton. Recall that the cell wall, okay, the cell wall allows for external support. It helps to keep the cell up, it helps to keep the cell rigid. Okay, so the cytoskeleton as well as the vacuole are components of the cell that helps to provide for support. The vacuole, sorry I didn't mention this earlier, but the vacuole will actually get turgid, get full of water, allowing for cell expansion, allowing for pressure from inside of the cell to keep the cell firm. Okay, allows for support interiorly. The cytoskeleton is generally a whole array of microfilaments, intermediate filaments, and microtubules, 3 different kinds of structures that help to keep, it actually is like a skeleton inside of the cell that helps to keep, the organelles in place in some ways, sometimes serves as tracks through which substances can actually move from one part of the cell to the other part of the cell, okay? So the cytoskeleton is also very important, and a lot of scientific research nowadays is focusing, okay, on the cytoskeleton, all right? Now the next part we're going to talk about is the nucleus. Okay, the nucleus sort of functions as the brains of a cell. Okay, it is a control center of the cell. This is where the information, okay, that actually accounts for the way the plant looks, the kind of chemicals the plant produces, and etc. This is where the master blueprint is held. Okay. All right. So inside here, inside of the nucleus, generally you'll have genetic material. Okay. Exterior to the nucleus, around the nucleus, you also have a double membrane, right? And in this double membrane, you'll have pores, okay, or holes so that allows substances to leave the nucleus sometimes, and it will actually allow things to get into the nucleus. Okay, so the nucleus is very important. It has genetic material inside. Okay, Another structure within the nucleus is called the nucleolus. The nucleolus is responsible for producing, let's see, ribosomal components, okay, ribosomal RNA. Remember, RNA is a kind of nucleic acid, all right? Also produces proteins that does contribute to the structure of the ribosome. And remember, ribosomes are places where proteins are put together. They're assembled there or they're synthesized there. Okay, so the important components of the nucleus include the double membrane that surrounds it, okay, that keeps certain things out, keeps certain things in, the nuclear pores, which allows substances to leave the nucleus, such as messenger RNA, which we're going to get into later in the semester, okay, which is important in terms of making proteins. The nucleolus, okay, as well. There's sort of like a little structure inside of the nucleus. It looks like a little round ball, okay, a spherical structure, okay, with a little bit of a gritty sort of texture to it, produces the ribosomal RNA and or other components of the ribosome. Then we have the genetic material. The genetic material in the nucleus, you can call it like the master blueprint. And the genetic material, of course, in plants is DNA, okay? Remember DNA, another nucleic acid as well as RNA, okay? So here, this is a very nice illustration. I like it when they use color this way too in the illustrations. So here you have the nucleus, okay? The nucleus and all this diffuse material in here, okay? This is called chromatin, okay? Chromatin. And this is the genetic material, okay, that will be used by the plant to make proteins eventually. Right? First, what happens is that the DNA, which helps make up the chromatin, okay, as well as protein. So chromatin is made up of protein and nucleic acid, yeah, DNA. And what this does, it will code for RNA. The RNA then will leave the nucleus through one of these nuclear pores, through the membrane, through the nuclear pores, and go out into the cytoplasm. And then from there, the messenger RNA will be used, and the ribosome will come together. And then what they'll do is they'll produce proteins. Okay. All right, we'll get into that more later, so I just wanted to give you a little preview. So here you have the nucleus, okay, with the nuclear membranes around it, and then here you have nuclear pores. Here you can see that the rough ER is contiguous, is actually contiguous with the membrane surrounding the nucleus. And here you have what looks like smooth ER, okay, which produces these substances, maybe lipids, maybe some other sort of substances. And then here you have the ribosomes where proteins are assembled, okay? And here you have the Golgi apparatus. Yeah, so here you can have a vesicle pinched off, okay, packaged within a membrane, then it goes to the Golgi body. So it actually moves through the stack, you know, going through this way until it's finally finished and refined. And sort of the Golgi apparatus is sort of like a refinery. It will take something from the ER, refine it, adapt it, you know, whatever it needs to be done, and then we'll export it, package it, and export it for export out of the cell. Okay, so all of these organelles are very important. Okay, very important. Now, talking about the nucleus. Okay, the nucleus, of course, is a place where you have the genetic material. Okay, so the next thing we're going to talk about is cell division, which includes mitosis and cytokinesis, and also the division of the cytoplasm, okay, which is called cytokinesis. Okay, so what happens during cell division is that what you'll start off with is a single cell, okay, and then after cell division, you'll end up with two cells. Okay, so usually the one cell that you start with is called the mother cell or the parent cell, and the two cells you end up with are called the daughter cells. Okay, it doesn't imply gender; it's just convention that we use it that way. Okay, so first of all, the cell generally goes through a process known as the cell cycle. Okay. And part of the cell cycle is called the interphase. The cell spends most of its time in the interphase, right? And one part of the interphase is called the gap 1 phase or the G1 phase, okay? The G1 phase is known also as the first gap phase. They call it a gap phase because when people were first investigating the cell and learning about this, okay, they thought that nothing much happened during the gap phases, okay, because we have a gap 1 phase, we also have a gap 2 phase, okay? So people thought that not much was going on during these times. But of course, we found out that's not true. During the gap 1 phase, the cell is actually getting larger, expanding, just like the vacuole is taking up water and it's actually making the cell expand, because the cell wall is a little bit flexible, it's a little bit plastic. It can actually get bigger. Okay, even though the cell wall generally is a rigid outermost part of the cell. So during the first gap phase, the cell is actually making more organelles, okay? It's growing, it's actually doing a lot of metabolic activities, right? Then after the gap 1 phase, okay, you have a phase known as the S phase, also known as it's known as the S phase because it's called the synthesis phase, okay, synthesis. And this S phase, or the synthesis phase, allows for the replication of DNA, okay, very important, right? That's why they call it the synthesis phase. The third part of the interphase is the gap 2 phase, or the second gap phase, okay, or the G2 phase. So during this time, the cell is readying itself to do cell division, to actually divide, okay? All right, so here we have an illustration of the cell cycle. So this very good coloration, you can see that the orange part that kind of circles toward the outside, this is the interphase. Okay, the interphase just means that the cell is not actively dividing, and the interphase can be divided into the gap 1 phase. Okay, and during this part, the cell is metabolically active. Okay, it's actually doing a lot of synthesizing of organelles. Okay, it's actually making more organelles. The cell is expanding and swelling, getting larger. Okay, that's what they call cell expansion. Then during the S phase, they call it the S phase because this is the synthesis phase where DNA is being synthesized. Okay. During the gap 2 phase, this is when the cell is, making more organelles, is also metabolically active, and it's readying themselves. They're readying themselves for mitosis, okay, or cell division. So during the M phase, this is during mitosis and cell division. So what you start off with here, you start off with a single cell. A single cell, right, going all the way through the interphase, through the gap 1 phase, through the synthesis phase, through the gap 2 phase, and then right after mitosis and cytokinesis, you will end up with 2 cells, 2 daughter cells from 1 parent cell. Okay? Now, so the cell cycle includes the M phase, or mitosis, and cytokinesis. Mitosis, strictly speaking, is what you call nuclear division, or the division of the nucleus, as well as the nuclear material. Okay? Cytokinesis is literally the division of the cell. Cyto refers to cell, and kinesis means splitting, or to cut. Yeah? All right, so here, cell division, okay, so this is a very good illustration because it's actually showing you what's happening with the chromosomes. So here, okay, at the beginning, if you were to actually have the genetic material in its condensed phase, or condensed state, which is known as chromosomal state, or chromosomes, here you see that you have 2 chromosomes in this cell, or in this nucleus, okay, so here you have 2 chromosomes. During the S phase, okay, so during the gap 1 phase, you actually have chromosomal material like this, okay, a single finger-shaped chromosome. Now, after DNA replication, what's happening during the S phase is all the DNA, okay, all the parts of this chromosome will actually duplicate itself. Okay, will actually make another copy of itself. So here, this is 1 single chromosome, okay? So here you actually have genetic material as well as proteins on this part here, which is called a chromatid, T-I-D, chromatid, yeah? And then this other one is its sister chromatid. So these 2 chromatids make up a chromosome, okay? After DNA replication, it's still only 1 chromosome. The same thing happens with this one that's colored a light blue. So this single chromosome, okay, will actually double its genetic material and proteins and make a sister chromatid. So here in this blue-colored chromosome, you have 1 chromatid and another chromatid. So each of these, this one here and this one here, should be identical to each other during DNA replication. We're going to go into DNA replication later in the semester, okay? So here you have these 2 chromosomes, each of them comprised of sister chromatids, genetically identical to each other. Now during mitosis, what will happen is you have this condensed state where you have these chromosomes, okay, condensed from the chromatin. Okay, so chromatin means colored threads. Okay, that's when the genetic material is very diffuse in the cell. Chromosome means colored body. Okay, I think they call it chromatin and chromosome because they were able to take a stain. So because remember, chromo means color, yeah, or colored. So here you have, you know, I guess it's not really blue necessarily, but when you can visualize it and see that it's condensed like this is called a chromosome. Okay, from decondensed or very filamentous and sort of like not really able to perceive single chromosomes, so you'll have chromatin. We'll look at that in a bit maybe, all right? So here, okay, after prophase, when the chromosomes are condensing from chromatin, then you'll have metaphase. A cell will go into a part called metaphase. And during metaphase, okay, meta means middle, maybe, okay? So metaphase, you'll have the chromosomes aligning themselves in the equator of the cell, okay? Then you'll have anaphase. During anaphase, you'll have the sister chromatids separating from each other, okay, with a spindle, you know, pulling them apart from each other. And then after, you know, after cell division itself, after mitosis and cytokinesis, you'll end up with two daughter cells, identical to its mother cell. Okay, these two daughter cells will be genetically identical to each other, okay, and also genetically identical to its parent cell. So this term here, it's supposed to be two diploid cells, yeah, but these two diploid cells, okay, what it means is that this cell here has two, you know, one set of two chromosomes. Okay, this one too has one set of two chromosomes. So we humans, we generally have 46 chromosomes in our body, right? Yes, we actually, before we're even conceived, you know, we sort of start, you know, you have a sperm from your father, an egg from your mother, okay, and what they'll do is they'll fuse during fertilization. So what happens is that the sperm and the egg actually has 23 chromosomes in each one, but after fertilization, okay, after unmentionable events, I'm just kidding. So after, you know, after fertilization, you'll have a diploid number. In the baby that's conceived, you'll have 46 chromosomes, whereas the sperm only had 23, the egg only had 23. Okay, fusing together to form a new individual, a zygote, with 46 chromosomes. Okay, same thing that happens in plants. I just use a human example because that is actually one that is more accessible to people sometimes when you think about it, you know? Alrighty, okay? Anyway, so cell division, okay, is comprised of mitosis and cytokinesis, okay, so during the M phase this happens, all right? The first phase is known as prophase. This is the first phase of mitosis, okay? During prophase, the chromatin, remember the chromatin or the colored threads, condenses and forms bodies known as chromosomes, okay? Another thing that happens is that you'll have two chromatids making up each chromosome. Yeah, two sister chromatids identical to each other. Okay? Another thing is that these two chromatids, okay, these two sister chromatids are joined at a part of the chromosome known as the centromere. Okay? And then during this part, okay, the nuclear membrane will break down, and the nucleolus that would have been there in the nucleus would not be visible anymore. Okay? Another thing that happens during prophase is that the spindle starts to form. The spindle will actually allow the separation of the sister chromatids from each other during anaphase. Yeah, so prophase. So here we have a nice illustration. Okay, black and white, nice illustration. Sometimes black and white illustrations can be the best because you can really see structure better. So here you see, this is a cell, okay, and here's the nucleus of the cell. What's happening is that the chromatin is condensing, okay, to form a chromosome. Another thing that's happening is the nuclear membrane is beginning to break down because during mitosis itself, generally you won't see the nuclear membrane. Another thing too is that generally here you see that you have a nucleolus. The nucleolus will kind of go into solution or become not visible, okay, during later phases of mitosis, okay? So that's prophase, okay, a depiction of prophase. You can also follow along in your textbook too, yep. when you're kind of looking at that. Okay, so in your textbook, they have illustrations of the cell cycle. And we also have on page 30, you'll have the different phases of mitosis, okay, illustrated there too. Okay, so generally too, the second phase of mitosis is known as metaphase. Okay, so during metaphase, the chromosomes will be arrayed at the equator of the cell or in the center of the cell. And another thing too is that the spindle will be more visible. The spindle begins to form during prophase, but the spindle becomes more visible in metaphase. Okay? So here we have a great photograph, you know. Here we have the colored bodies, okay, or the chromosomes, yeah. And what you'll see here is that this is a single chromosome with two sister chromatids, and these two sister chromatids are joined together at a place called the centromere. Another thing that you see is you see all these, you know, separate chromosomes. You see that you can see the spindle formed here. Okay, very good. This is an excellent photograph. Okay, so here you have, they probably stained this cell, yeah, so where you would be able to see the chromosomes. That's where they got the term chromosome from it being able to take a stain. So you have the spindle here, and here you have an illustration that shows it also too. So this cell here happens to have 4 chromosomes. Okay, so this, you can see here, you see one sister chromatid, another sister chromatid of this chromosome joined together at the centromere, and then here you see the spindle, okay, that will connect, okay, to the centromere. All right. Very excellent illustrations of metaphase. The next phase after metaphase is called anaphase. Okay, so anaphase. During anaphase, the basic thing that happens is that the sister chromatids will separate and move to either side of the cell. Okay, and here is a good illustration of anaphase as well. So here you can see that this is a sister chromatid to this one here. But what they're doing is that they're being pulled apart, okay, pulled apart as these, the spindle is shortening, the filaments are shortening and shortening. What they're doing is they're pulling the sister chromatids apart. Once the sister chromatids are apart from each other, they will be known as chromosomes, no longer chromatids. So here you have a single chromosome, and here you have another chromosome. So these two chromosomes should be identical to each other, okay, in terms of the DNA sequence and the proteins that they contain. They should be identical to each other. Here's another pair, okay, of chromosomes, single chromosomes, once again. So in this cell here, you will end up with, okay, right, so here we have mitosis and cytokinesis, and then we have telophase or telophase, okay? So generally during telophase is almost the inverse of prophase. What happens in the chromosomes will get into their diffuse state known as chromatin. Another thing that will happen is that the nuclear membrane will begin to reappear and reform, right? And then here we have, okay, this is another excellent photograph. Okay, so here you can see that the genetic material, these chromosomes, are starting to diffuse back into chromatin. It's a little bit harder to distinguish individual chromosomes from each other. Another thing we see is that here you can see the nuclear membrane reforming, okay, around the genetic material, all right? And so, and in this cell here, and you can also see that these two daughter cells, okay, are separating each other because during telophase or telophase, you have the beginning of cytokinesis or cell division. Remember up till now, prophase, metaphase, anaphase, and telophase, okay, these 4 parts are parts of nuclear division or the division of the nucleus, right? So during cytokinesis, you will actually have the division of the cell. In plants, okay, cytokinesis, okay, cytokinesis will be the division of the cytoplasm. And in plants, what happens? You have the formation of something called the cell plate, a little bit different from animal cells. In animal cells, generally the cells will pretty much almost like pinch in half, okay, sort of pinch together like this and pinch in half. What will happen with plant cells is generally you'll have, you know, the two daughter cells are still joined together. You'll have the beginnings, you know, you'll have these components, lipids and such, aggregating between the two the two parts that are going to be two daughter cells. The fragmoplast will accumulate then, that will form something called the cell plate. What then happens is that the cell plate will then develop into the cell walls that will separate the two cells from each other, okay, with the middle lamella between them as well. Okay, anyway, so let's see. So here, okay, we see again, that very good illustration showing at the beginning of the cell cycle here, during the gap 1 phase, okay, you generally, in this cell, you have 2 chromosomes. So here you have one single set of 2 chromosomes, or the 2N number, or the diploid number is 2, okay? All right, then what happens is that during the S phase of the interphase, you'll have replication of the DNA. So the DNA making up this genetic material here will duplicate itself, okay, during DNA synthesis. So what you'll end up with is 2 sister chromatids making up a single chromosome. Same thing with the blue one. Then in metaphase of mitosis, okay, you'll actually have here, all right, you'll have here the 2 sister chromatids, and this other chromosome too, they're kind of aligned in the middle of the cell, okay? And then during anaphase, you'll have the sister chromatids separating. Now they are single chromosomes, all right? Single chromosomes separating from each other, okay? And then now they're single chromosomes, all right? And at the end, after telophase, with a reformation of the nuclear membrane, okay, the genetic material will go back into its diffuse forms. What you'll end up with 2 daughter cells. And in these 2 daughter cells, you have a diploid number, 1 chromosome from its father, 1 chromosome from its mother, okay, starting from here. And then what you end up with, you have 2 chromosomes into each of these cells. And these 2 cells should be genetically identical to each other, as well as being genetically identical, okay, to its parent cell that it started with, okay? Right, let's see. Okay, let's see then, okay? So, one of the last things we're going to talk about today, okay, we're going to talk about, the reason, why do we do mitosis? Well, what are the functions of mitosis? Okay, so why do we do this? For one thing, what do plants do? Just like any living thing, plants grow, okay? So plants will actually grow, and generally active cell division happens in the plant. This is a little preview for our next lecture, okay? This is where plants grow from. They grow from tissues known as meristems, okay? So why do mitosis? Okay, we've done it too from the time when we're very, very little. For one thing that happens, of course, when you're born, you're a little baby, right? You're a little baby. What happens inside of your body too, as you're maturing, as you're growing through your childhood years into adolescence and everything like that, into young adulthood, and even as adults, you keep growing up to a certain point. I know sometimes human beings, you might think, well, 18, 21, you're an adult, but some people still actually grow taller after that, we still grow. So growth is one major part of mitosis, is growth. And in the plant cell, of course, okay, the plant cells will actually have mitosis to actually form things like this leaf. All right, so this leaf is a result from cell division and mitosis and cytokinesis, which comprise cell division. All right, Miley-scented fern, very important. Okay, another function of mitosis too, okay, is that repair. What happens is that, in plants as well as in animals, they get injured as well as we get injured. You might not think about it, but if you actually have a plant indoor, say you have a very large Ficus benjamina, one of those ficuses in your office, or if you have a spathiphyllum on your desk or something like that. If you do have like a large plant in a corner, or an areca palm in a corner of your office, and somehow people brush past it every time they go by, you're actually damaging the plant a little bit. Did you know that? That every time you brush past it, you're actually wounding it a little bit. So in plants, what happens, so another thing that can happen, somebody might come and they might actually take a plant, people fiddle sometimes when they're nervous, or if they just have a lot of nervous energy, say if they had too much coffee that morning. So what people will do sometimes, they'll actually take a plant and they'll break off the leaves, just like you're sitting on the grass, and you kind of pluck at the grass kind of growing near where you're sitting, right? Sort of like a nervous tick. So what you're actually doing, you are actually damaging the plant. But what the plant does is, just like in our bodies, you know, when we get a cut on our finger, or we're playing basketball and we trip and then we fall and skin our knee or something, okay? That's wound tissue, right? So on our tissues as well as plant tissues, they will actually do mitosis to repair any damage that has happened to them. Okay, so that's a second function. So we have growth, and we also have repair that happens in our bodies as well as in plant bodies as well. Very important. So mitosis, of course, is essential. Pretty much everything that we talk about in this class really is essential to the life of the plant. Okay. Another thing plants can do, okay, that we humans are not really able to do so much anymore, is they can do asexual reproduction. Recall that before we mentioned that sexual reproduction has to do with having two parents, right? So of course animals generally mostly can only do sexual reproduction. So sexual reproduction has when you have two parents. For myself, of course, my father and my mother. Yeah, they actually both contributed genetic material to make me. Yes, they also contributed genetic material to make my sisters and my brother. But of course, we sort of look like a little bit alike, but of course we're not identical. We're going to get into that later when we talk about the flower and sexual reproduction, or meiosis. Another thing too is that plants, amazingly, not like us, they can do sexual reproduction, but they can also do asexual reproduction. I'm showing this kalo plant again, okay, or this kalo leaf, because kalo is a plant that is usually propagated mostly through asexual reproduction. Okay, so you can imagine this leaf, kind of sticking out of the water in a lo'i, a lo'i kalo. A lo'i is usually usually where people grow kalo nowadays, yeah, which you have water flowing in, and then you'll have at the end of the lo'i water flowing out again. There's whole philosophies, about growing kalo and its place in the society and the way to care for it, how to grow it, and etc. Anyway, so the leaf is generally attached to a stem. Okay, and the stem part of the kalo is called the corm, and this is the part that people use to make poi from. All right. So when they're propagating the kalo plant, they usually do it through asexual propagation. They'll take part of the leaf stem, the leaves sticking up, part of the stem, okay, in something called a huli, and then what they'll do is they'll cut off the rest of the corm. They'll use part of the corm for either cooking and eating it as is, or they'll use it to pound it with a little bit of water, and they'll make a substance known as pa'i'ai, okay, which is a stiff, kind of firm form, And to make poi itself, you'll actually put the pa'i'ai, and little by little, you add water little by little, okay? And then what you'll do is that you actually make it a little bit more watery. Sometimes people like to eat poi that is very, very thick, and that usually people refer to as one-finger poi. Okay. If you thin it a little bit more, they call it two-finger poi, because of course Hawaiian people in the olden days, people mostly ate with their fingers. Okay, and sometimes they'll use like a tea leaf like a plate to hold their food on. Okay. Anyway, I just show the kalo because this here is a plant very important to Hawaiian culture. Also a plant notable because it's one of the plants that you do asexual reproduction with usually. I know nowadays too, people are also doing sexual reproduction when they're trying to develop new forms of kalo plant. And now that is actually an area also of controversy nowadays, fairly recent controversy, because I know that there's a little bit, some scientists feel that they should be able to patent life forms. So that pretty much was a kind of controversial subject, but it's still very interesting. So of course, plants are very interesting in terms of its, the different ways that they affect us, whether culturally, in case of the kalo, and even politically. Okay, such as some things that we might talk about, such as sustainable agriculture. Okay, now why don't we go to the Elmo? And we can review a little bit of something that we talked about a little earlier. Try to review parts of the cell, okay? So here we have a nice model of the cell, and a plant cell. And for one thing, okay, we can see that the exterior part of the cell, of course, is a cell wall, Okay, we even have a little bit, it's not easy to see here, but this part here might actually stand for the middle lamella, which is the part that kind of links this cell with a cell that might be adjacent to it, right next to it. Okay, so here we have the cell, the middle lamella, then we'll have the cell wall. And in this cell, it only has a single cell wall or primary cell wall. Another thing we can see with this model is you can see the cytoplasmic connections between this cell and the cell that would be adjacent to it, okay? Also known as the plasmodesmata. Remember plasmodesmata, or the connection between this cell and the cell that would be next to it, all right? So we have the middle lamella, we have the cell wall, then we have the cell membrane interior to that, okay, which is pretty much like a film of oil. It's very, very, very thin, okay? The cell membrane mainly composed of phospholipids and proteins, yeah? Remember protein icebergs in a sea of lipids, all right? Then we talked a little bit about, well, we didn't really talk about it so much, but we also have the cytosol, or the fluid inside of the cell, in which you have the organelles suspended. And of course, one organelle we talked about, the first one we talked about, was a chloroplast. Yeah, and generally when you look at a model of a plant cell, you can see that the chloroplasts, of course, are generally colored green. And here we have cutaways of the chloroplasts, okay, showing the stacks of thylakoid membranes, and that will be the greenest parts of the cells because, of the organelle, because in the grana, in the thylakoid membranes, you have embedded in them photosystems. And photosystems, okay, are made up of hundreds of pigment molecules, of chlorophylls and carotenoids, and etc. Okay, so we have the chloroplast. We also have so the chloroplast has a double membrane around it, remember? The mitochondrion, okay, which are these kind of usually colored red when people have a colored photograph or a drawing. I guess because you can see and distinguish them from the chloroplast. So if you look at a cutaway of this, you can almost sort of see the little, cristae, the inner membrane, okay? So if the chloroplast is the organelle that does photosynthesis, you can remember that the mitochondrion, which also has a double membrane, okay, it being the organelle that is specialized, okay, to do cellular respiration. So inside of the plant cell, you have like a marvel, okay, a marvelous, sort of wonderful, delegation of function to different structures inside of the cell. So the chloroplast, of course, does photosynthesis. Mitochondrion does most, okay, of cellular respiration, producing the most ATP. We also have here, let's see, the ER, okay, I'm sorry, let's go to the vacuole first. We have the vacuole, and here you can see in this cell, at least, it's very large. The vacuole has cell sap in there. All right, here we even see little crystals, okay? See crystals, remember we talked about calcium oxalate in the vacuole of kalo plant, yeah? So these crystals, are, some people don't know what these are for. In fact, most people don't know what they're for. Neither do I. But anyway, these crystals, they can actually be occlusions in here that might be, crystallization or precipitation of substances inside of the vacuole that maybe the plant doesn't need. Other components that might be in a vacuole might include anthocyanins. Remember that pigmented, that kind of purplish, pinkish colored onion cells that we looked at a little earlier? Okay, they also have salts, and they have proteins floating around in here, sugars in here too. Vacuole, very important, not just for the things that it contains, but also for the fact that it provides for internal support of the cell. Okay, let's see, we also have the membranous organelle known as the ER, or the endoplasmic reticulum. Okay, we have the rough ER studded with ribosomes. We also have the smooth ER that generally mostly produces lipids, with the rough ER producing proteins. Then we have something like the Golgi apparatus, okay, where the products that are produced by the ER, okay, is actually finished, packaged, and then packaged again for export out of the cell. Okay, another thing we talked about was the nucleus of the cell. Here, this is a good nucleus. It's not really spherical, but you can see that it's a little bit squished because the vacuole is actually pushing it against the side of the cell. So sometimes when you're looking at a photomicrograph of a cell, you actually see that the nucleus is actually squished over to the side. Okay, so here you can see the double membrane around the nucleus. Here you can see the nuclear pores. All right? And inside of the nucleus, you'll have the genetic material in its diffuse form, like this, in chromatin. Then here you have the structure known as a nucleolus, which will provide for and produce the components making up the ribosomes, because you remember ribosomes are very important in terms of making proteins, okay? They're actually places where the cell is actually assembles proteins, okay? All right, so here, okay, we can actually finish up with a summary. Okay, so we talked a little bit about the components of the plant cell. Yes, we did a little review of a couple of the organelles, the chloroplast and the mitochondrion, and then we talked about the other parts of the plant cell, including, let's see, the vacuole, the membranous organelles, the ER, the rough ER, the smooth ER, the Golgi apparatus. We talked about the nucleus and the components of the nucleus. We also talked about what these parts do. Okay, so every single part of the plant cell does something. They're all necessary. Nature abhors a vacuum, they say, but nature also doesn't like waste either. Nature pretty much you won't usually have something inside of a cell unless it's used for something. Okay, nature is among the most economical sort of forces, okay, in the universe as far as we know. We also talked about the cell cycle, the interphase, okay, the interphase and the M phase, all right? Interphase being comprised of the gap 1 phase, the S phase or the synthesis phase, and the gap 2 phases, yes? And of course, all these are necessary. These are all necessary in the life of the cell. We also talked about mitosis, okay, or nuclear division. Also, cytokinesis, the phases of mitosis, prophase, metaphase, anaphase, and telophase. And we talked about cytokinesis and the processes, that occur during these phases. Okay. We also talked about the functions of mitosis, including growth, okay, which pretty much all living things. Recall that when we talked about this before, that everything that's alive does grow. Everything that's alive has a capacity to grow. Remember that growth is an irreversible increase in size. Okay, so the functions of mitosis includes growth, which all living things can do. Okay, they also have to do with repair when something gets injured, whether it's a plant that somebody's picking apart or it's ourselves. If we're chopping onions or something, making spaghetti, chopping onions and garlic and nicking your finger a little bit. Okay, and also mitosis functions also in asexual reproduction that plants can do as well. Okay, anyway, the next time we'll be talking about the plant body. All right, so I'm glad that we're here together once again, and looking forward to the next time we see each other. So that's all we have for today, and we'll see you next time. Okay, aloha.